The information in this module is accurate and complete to the best of our knowledge. All recommendations are made without guarantee on the part of the author or the sponsoring institutions. The author and the sponsoring institutions disclaim any liability in connection with the use of this information.
Establishing similarities was mentioned as one of the bioinformatic challenges in the previous lecture. Indeed, the notion of similar sequences is fundamental in the analysis of all genetic data. This presentation briefly discusses why this is so, along with some issues related to the establishment of similarities by computational means. Describe the notions of homologous, orthologous, and paralogous sequences. Recall the types of sequence mutations and the main phenomena that produces them, and explain the features of a sequence alignment. Biological homology means likeness in structure between parts of different organisms that is due to evolutionary differentiation from a corresponding part in a common ancestor. These homologies can be traced to similarity of nucleotide or amino acid sequences. Some, but not all, the observable similarities in genetic sequences originate in biological homologies. The search for homologies at molecular levels starts with string comparisons. The table compares the segments from positions 120 to position 162 of the protein H1 in different species. The comparison shows a remarkable similarity, which may be taken as a signal of potential homology. The characters that are not repeated in all segments are marked in red. It is worth remarking that the differences in the characters occur all in a few well-defined positions. This table compares in turn segments of the PAX6 gene, which plays an important role in the formation of tissue and organs during embryonic development. The segments are taken from different species, and although the comparison indicates a high level of similarity, the differences in characters occur in a less organized manner than in protein segments. Where do these similarities come from? The short answer is molecular evolution. At genetic level, sequence homologies are often interpreted as a signal of a common ancestor. A homologous gene, or homolog, is a gene inherited in two species by a common ancestor. They are classified as paralogous or orthologous. Paralogous genes, or paralogs, are homologs that result from a duplication event. Orthologous genes, or orthologs, are homologous that result from a speciation event. Gene information may be passed either through a duplication event or a speciation event. Usually, a duplicated gene evolves a new function while a gene passed to a new species tends to retain its function. In this hypothetical sequence of events, a speciation is followed by a duplication event. The first produces the ortholog gene 1 and 2. Then gene 2 undergoes a duplication event that produces two paralogs. These paralogs are still orthologs of gene 1. The same nomenclature applies in case that a duplication event precedes a speciation event. In this case, a duplication event generates two paralogs of gene G. Then the speciation event generates ortholog copies of each of the paralog pairs of genes. As illustrated with the comparisons of protein and gene sequence segments, orthologs and paralogs are far from being perfect copies. Besides speciation and duplication, a wide variety of cellular events produce character substitutions, character deletions, or character insertions in a sequence. These cellular events must be taken into account when assessing the similarity of two sequences. To further complicate homology detections, not all similarities in sequence translate in similar functions or structures. This is, sequence similarity is not synonymous of biological homology. Copy errors is a main source of character substitution. When a cell divides, it makes a copy of its DNA. Sometimes the copy is not perfect substituting. Thus, one or more of the characters of the original sequence. DNA is broken down by the action of natural or external agents. When this happens, the cell repairs the damage. Nevertheless, just as DNA copies, cell restoration is not always perfect, and the repaired DNA might end up differing in a few characters from the original one. Insertions and deletions are produced by a variety of cellular events as well. One of them is gene translocation. A gene translocation is an exchange of segments between non-homologous genes. This exchange may or may not reverse one or both segments, and may be balanced, meaning that it does not affect gene functionality, or unbalanced, if it affects it. Unbalanced translocation is a source of genetic illness. Unequal crossing over refer to instances when unequal pairs of homologous chromosomes mix mispaired segments. In such case, one of the resulting segments will have an extra copy of the duplication and the other will be a missing copy. Transposons or transposable elements are sequences that move around the genome. There are three main different mechanisms for transposition. Conservative transpositions, in which the element itself moves from the donor site into the target site. Replicative transposition, in which the element moves a copy of itself to a new site via a DNA intermediate. And a retrotransposition, in which the element makes an RNA copy of itself, which is reversed, transcribed into a DNA copy, and then inserted. Here is an example of two sequence segments that come from homologous sequences. Indeed, hemoglobin A and B are proteins that carry oxygen to organs and tissues and transports carbon dioxide from the organs and tissues back to the lungs. Can we ascertain this homology with a sequence similarity analysis? It all reduces to one central question. What is the origin of the mismatched? We have two options. Accept the mismatched as a character substitution, or reject the mismatched by inserting a gap symbols and shifting the sequence as shown in the second comparison. As a rule of thumb, we take a combination of substitutions and insertions and deletion that maximizes the number of matches. Such combination assumes that most characters are conserved by the action of evolution. We can only guess the transformation events that produce the differences between HBA and HBB. This diagram shows one particular selection of events out of a large number of possibilities. The selection preserves as many matches as possible and makes a balanced trade-off between substitutions and insertions and deletions. Of course, this is just a conjecture. The conjecture is translated to alignments by expressing insertion and deletions with a gap symbol. In general, the alignment of two sequence, or pairwise alignment, is a two-row array that summarizes a particular choice of evolutionary transformations that take two sequences apart. This conjecture may prove or disprove the hypothesis that the sequences are homologous. Assume that we have an appropriate scoring scheme for a particular alignment. Still, we have an exponential number of possible alignments. We must search among them for one that maximizes similitude. Such optimal alignment cannot be obtained without adequate decisions on how to treat a mismatch. This is, adequate decisions between substitution, insertions, or deletions. To summarize, we are presented with two important dilemmas. What is the appropriate scoring scheme? And whether there is an efficient way to find an optimal alignment. An answer to the first question is provided by the substitution matrices, which are discussed in the next module. And in subsequent modules, we examine some algorithmic solutions to the problems of finding an optimal alignment.